Hello everybody, welcome to today's live broadcast Key Considerations when Selecting mRNA CDMO Partner. I'm Chris Spivey, the Editorial Director of Pharmaceutical Technology, and I'll be the moderator for today's event. We're extremely pleased to bring you this webcast presented by Pharmaceutical Technology and sponsored by Samsung Biologics. I'd like to share a brief statement from our sponsor. Samsung Biologics is a fully integrated CDMO offering state-of-the-art contract development and manufacturing services. With proven regulatory approvals, the largest capacity and the fastest throughput, Samsung Biologics is an award-winning partner of choice and is uniquely able to support the development and manufacture of biologic products at every stage of the process, while meeting the evolving needs of biopharmaceutical companies worldwide. We have a few important announcements before we begin. This webcast is designed to be interactive, and we encourage you to ask questions during the event. You can submit questions by typing them in the Q&A box, which can be found at the bottom of the video player. You can enlarge the slide window by clicking on the small icon in the bottom right corner of the media player, and the slides will advance automatically during this event. But if you have any technical problems viewing or hearing this presentation, please click on the question mark Help widget in the top right of your presentation window. And I'd now like to introduce today's speakers. It's a great pleasure to be joined today by Pierre, Catignol, and Eunseo, NC Lee. Pierre is the Executive Vice President and Head of Manufacturing at Samsung Biologics. He is a recognized industry expert with a demonstrated history of 26 plus years in the biopharmaceutical and pharmaceutical industry. NC Lee is a senior scientist of mRNA TF at Samsung Biologics. After joining Samsung Biologics in 2021, she has led the tech transfer project in both lab and at-scale lipid nanoparticle manufacturing processes. So thank you both very much for joining us today. Pierre, could you please get us started?
Thank you very much. So the presentation will have two main chapters. The chapter 1 will be a review about general mRNA manufacturing considerations. And the chapter 2 will be a review about Samsung Biologics capabilities. So let's start with chapter 1.
Hello, this is Eunseo and Lee, and then I will go over chapter 1 on mRNA manufacturing. So let me start the presentation with the challenges of in-house biomanufacturing. In general, it takes a significant time and money to build large-scale facilities. Securing highly skilled workforce in a broad range of specific technologies, including a process development, analytics, and regulatory affairs can be competitive. Also, biomanufacturing market is often difficult to forecast its dynamics due to fast-developing technologies and unexpected situations like COVID-19 pandemic. Then, how about the challenges in the mRNA market specifically? There's no doubt that the COVID-19 pandemic has accelerated the clinical efficacy of mRNA-based therapeutics. Since this area has just been commercialized after the emergence of pandemic, when you take a closer look in this field, mRNA CDMO company needs to understand the challenges from the product developer in research level first. From the perspective of product developers, mRNA is very fragile inside of the body, leading to failing in full protein expression. To prolong the gene expression, vector design control is very important. Commercially available lipid nanoparticles, or we call LNPs, mostly accumulate in the liver, so it greatly lacks target specificity. Also, tissue tropism and delivery efficiency of LNPs triggers scientists to look for a novel type of cation lipid. The intrinsic immunogenicity of LNP is another major issue to be resolved. Even though researchers put a tremendous effort to improve these problems, there are very limited commercial sources for a potential manufacturing. While the forefront technologies have been developed to face these challenges for drug developer, mRNA CDMO companies should be able to provide the optimal solutions. First of all, establishing the advanced quality requirement is necessary to transfer research-grade reagents into critical raw materials. This quality control should be further expanded to drug substance and drug product to assess the quality attributes on each. Critical process parameters should be comprehensively evaluated to keep the consistency and controllability of the production. Depending on the drug candidate, a desired amount of production and processes can be greatly varied. Scalability and flexibility of production comes into consideration as well. And at the end, integrating the fragmented mRNA supply chain across the continents will relieve the complexity of handling the mRNA-LNP drug substance. To overcome previously addressed challenges in the field of mRNA-based therapeutics, there are 4 key considerations: agility, capability, technical expertise, and quality when it comes to selecting a mRNA CDMO partner. So from next slide, I'm going to explain in detail what makes each attribute so important, especially in manufacturing mRNA-based therapeutics. Overall, mRNA therapeutics bring all different elements together, from RNA payload engineering to manufacturing process engineering. As mentioned earlier, mRNA CDMO companies need to understand what kind of services are in thirst from RNA product developers. So RNA CDMO is not just on S-scale mRNA production, but science-driven service. Optimizing a platform to evolve the current mRNA-based system in the research level necessitates the understanding of RNA molecular and payload engineering. For example, RNA payload can be categorized into non-replicating and replicating constructs. The best example of non-replicating construct is messenger RNA, but the reason for developing other types of non-replicating construct is to find an alternative approach to the current capping agent, such as Vaccinia enzyme and CleanCap. Either applying a new type of capping agent on mRNA or finding a novel RNA type, such as circular RNA, can be introduced under this category. Replicating constructs such as self-amplifying RNA or saRNA has been actively studied to relieve high dosing and repetitive injection requirement. Along with RNA payload, there are a number of combinations of lipid derivatives in the pursuit of optimizing an LNP delivery carrier to overcome immune response or tissue specificity. Also, scientists try to conjugate protein peptides on the surface of LNPs for a targeted delivery. So when modalities on various types of RNA and delivery carriers are well explored, a comprehensive manufacturing process comes afterward. Thanks to a relatively simple cell-free production of mRNA over conventional biologics, multiple RNA payloads might be accommodated from a general RNA manufacturing process platform through in vitro transcription. While T-mixing system is generally adopted for at-scale production, microfluidics or advanced cross-flow systems can be applied per client's demand. If mRNA CDMO company fully grasps all these systems on LNP formulation and executes comparability study, it should be able to flexibly provide a diversified scale-up regardless of the initial platform for a small-scale study. Evaluating mRNA-LNP drug substance under validated analytical method is another crucial attribute of mRNA CDMO company. So with significant advances in the engineering of RNA payload, delivery carrier, and manufacturing, the most important asset from mRNA CDMO company would be technical experts. Highly experienced staff with hands-on experience will ensure the operational excellence and quality consistency. Therefore, not only the advanced talent acquisition, but also training the manpower will hold promises for more access to manufacturing capability. The overall mRNA manufacturing processes begin with the plasmid DNA or pDNA, generation containing a vector designed for mRNA construct. The pDNA is linearized to transcribe the mRNA and subsequently degrade by DNase. In vitro transcribed RNA is further purified and encapsulated inside of the lipid nanoparticles. To offer multiple modalities from numerous clients, manufacturing flexibility in various types of RNA payload and LNP formulation technique is a key component for an immediate responsiveness. So let's take a look into these in more detail from the next slide. Generally, an mRNA core technology to generate a sufficient quantity of the encoded gene comes from a design of mRNA construct. Therefore, mRNA CDMO must understand the molecular mechanisms in mRNA construct. Designing this factor incorporates 5' cap structure, 5' and 3' untranslated region, coding sequence, and poly-A tail. Coding sequence design can be based on literature and open sources data such as Eterna. Since billions of sequences can be augmented in the optimization of coding and non-coding sequences, AI is a rising star in a targeted design to predict the durability of drug from the secondary structure analysis. Then, in silico screened sequences are inserted into the DNA template and in vitro transcribed into mRNA to confirm the functionality of candidates at both in vitro and in vivo. After all, the most optimal RNA sequence will be selected to satisfy the standard of stability, structure, gene expression, and immunogenicity. Most of product developers secure on mRNA or prefer CDMO companies in geographical proximity with highly experienced mRNA experts to develop a novel RNA. When developers use on mRNA, CDMO company may not be able to access the payload information due to intellectual property protection. However, a considerable focus is essential with regards to potential impact on immunogenicity, drug efficacy and purity, manufacturing productivity, and operational expense. In order to successfully manufacture the drug substance, a deeper understanding in both product and process is necessary to prove platform versatility. The beginning of RNA process design is to define quality target product profile, or QTPP. Example of QTPP is dose, sequence, organ target, administration route, and payload type. Then process modeling in each unit operation further design the manufacturing process. The examples of process modeling in vitro transcription is RNA polymerase reaction, type of capping, byproducts, and impurities. For mRNA purification, membrane capability, resin load challenge, and impurity clearance are included. Lastly, LNP mixing and tangential flow filtration model, target LNP size and whole dispersity index are applied on LNP formulation and purification. Choosing the right equipment for an appropriate process design is crucial. So for mRNA CDMO company, it is important to be well aware of various equipment available in the current market in the unit operation. This means that catching up the future technology innovations in a related field can add up the list, such as an advanced cross-flow equipment from the previous slide to formulate the LNP in aseptic and low pressurized condition. At the early stage of process development, identifying process parameters are required to draw a new roadmap on versatile RNA platform. Here, you can see the list of process parameters in each unit operation. Defining QTPP, process parameters, and process modeling is a part of quality by design, or QBD approach. This QBD approach greatly leverages prospective and refinement on mRNA-LNP manufacturing process for a tuned product. One important underlying principle of QBD is to directly connect quality to process for pharmaceutical safety and efficacy. Critical quality attributes or CQAs serve as the basis of process development, and the potential CQAs for RNA drug substance and drug product are listed here. CQAs are often in accordance with the analytical testing scope and drug specification. In this sense, assessing critical quality attributes under validated analytical method is to best move forward as a CDMO company. While a procedure from analytical method development to product characterization may be equally applied in providing analytical service as we did in conventional biologics CDMO, a new analytical method such as residual abnormal measurement in LNPs may be introduced specifically for mRNA-LNP drug. Or it is possible to suggest an alternative analytical method through process analytical technology systems to evaluate the CQAs. Therefore, an enhanced quality attribute will control the shape of process development and eventually maintain a high level of product quality to assure a key element of mRNA CDMO. As manufacturing mRNA-based therapeutics is composed of a complex set of processes from pDNA linearization, in vitro transcription, mRNA purification, LNP formulation to final seal and finish, integrating all these processes into one site through end-to-end service can greatly satiate the demand from product developers for a high-quality assurance. Fully integrated CDMO service will utilize the optimal asset, guarantee a fast supply, and reduce the cost of goods. Below is the general scope of end-to-end CDMO service. It starts with product, process, analytical development, and non-GMP, DS, and DP production and testing. Then GMP, DS, DP production test, fill and finish, labeling and packaging, quality control and biosafety testing, then finally warehousing and distribution. To CDMO company, one of the important factors is to show the agility by reducing the overall production time. For example, in a product development stage, CDMO company may consider a partnership with biotech in designing an optimized plasmid DNA vector or finding the best lipid components for a specific target delivery. Also, outsourcing raw materials such as plasmid DNA and lipids can complement a potential supply shortage. Once the mRNA drug product is ready, finding a distributor of cold storage units can be challenging. So this stage is where having a partnership becomes important to relieve the innate limitation of a current mRNA drug product. Again, key CDMO personnel should be expected to be experts in RNA technology with CDMO manufacturing experience, particularly in large-scale process development. They rely heavily on a CDMO partner for expertise in development, manufacturing, scale-out, and analytical capability. Through the end-to-end service, mRNA CDMO companies should be able to read a rapidly changing market needs and to be ready to fulfill public and industry demands when required. For a CDMO company, a robust manufacturing with clear scalability is a key factor, especially in tech transfer. In a recent case of Samsung Biologics, Greenlight's mRNA synthesis reaction had a titer of 12 grams per liter at a commercial scale and produced 650 grams of mRNA. This titer was comparable to a small-scale production. When you also look at two critical quality attributes of LNP size and mRNA encapsulation percentage, you see that the results from small and large scales are comparable as well. In this case, mRNA synthesis and LNP formulations were achieved without the need for customization using the standard equipment for mRNA CDMO. So the next one is the last page of the chapter. In order to build a new process from a specific class of RNA, product, and modalities, mRNA CDMO company needs to be open-minded. Here, finding a right partner would be strategically helpful to build up a robust internal process development from the scratch. Samsung Biologics recently invested $50 million on a drug delivery platform of deep molecular profiling and advanced analytics to natural nanoparticles and information molecules. This partnership will lay the groundwork for establishing a platform of mRNA technology and expand the service scope as a CDMO company. So I'm going to wrap up my chapter and hand over to Pierre.
Thank you very much, Eunseo. So now let's have a look about Samsung Biologics' capabilities regarding mRNA services. So first of all, let's review the scope of our services. So we can be your strategic partner for process development, tax manufacturing, phase 1 and 2 scale-up, and manufacturing, phase 3 and commercial scale-up, and corresponding manufacturing. To support all this full scope, we have on site in Korea a MSAT lab with various related equipments at small scale from 100 milliliter to up to 10 liter. And we have GMP manufacturing facility, large scale, with all related equipments. And in this facility, we can produce up to a 200 liter scale batches. So we started cGMP mRNA drug substance manufacturing last April 15 in 2022. And we now offer a full end-to-end service package from pDNA manufacturing. So these steps will be available in Samsung Biologics early 2024. And then for mRNA drug substance manufacturing, so including pDNA linearization, mRNA synthesis, mRNA purification, LNP formulation. We are doing a drug product too, with fill and finish services, with liquid or freeze-dry forms, and labeling and packaging, QC testing, and QA batch release. So we have, as you can see on the slide, a very large equipment library that gives us the capability to produce different scale for mRNA drug substance and mRNA encapsulations. So we can produce from milligrams for preclinical and phase 1, up to grams for clinical stage, up to hundreds of grams in, that means phase 3 commercial scale in a GMP manufacturing facility. So we have 2 mRNA laboratory that is already in operation. So this mRNA laboratory allows us to leverage our great experience that we have make acquisition during the last 10 years in tech transfer, process development, and characterization. Mainly, all these competencies have been acquired for monoclonal antibody. So regarding technology transfer, we can do all kinds of services. So IVT, LNP process transfer, we can do gap assessment, lab scale verification runs, all cleaning, verification and validation related studies, and non-GMP analytical testing. Regarding process development, we can do IVT and LNP process design, IVT mixing studies, LNP formulation studies. We have chromatography, resin, and filters, and we can do analytical method developments. And regarding process characterization and process validation, so we have a scale-down model development that is qualified. We can do the process validation master plan, all kinds of PC studies execution. We can run a risk assessment for you and do PC reports generation. So we have various number of equipments to fit your needs. So this lab has been opened last October 2021 and is fully operational. As a leading company, our core strength comes with our people. So we have top-class talent continuously joining Samsung Biologics, enabling us for all future multi-modality challenges. This incredible growth has been supported by attracting the best people on the market and a unique system of training to quick onboarding and to make the skill acquisition. So Samsung Biologics is ranked in Korea #1 most influential biopharmaceutical company. The people working for Samsung Biologics have 55% bachelor and higher degrees, 70% coming from the top 7 universities, and the turnover we have is around 4%. That allows us to capitalize a lot on our people. On the graph, you can see the very high growth we have handled during the last 10 years, from 100 employees to approximately 4,750 people. This is the growth we have been able to manage with attracting the best people and training them to onboard quickly and make the skill acquisition. On top of attracting the best people, our track record and history has brought us to the highest level of compliance. And regarding mRNA in particular, we have mRNA dedicated teams and experts to answer all your questions and taking care of your product. We have full scope of services, and we have all flexible scales from a laboratory to commercial. And we can tailor according to your needs. So we have a proven quality track record at the highest standard with more than 150 approvals from over 25 health authorities from our history of monoclonal antibodies. So thank you very much for your attention and for your interest. Thank you.
Well, thank you, Eunseo and Pierre, for such informative and detailed presentations. Critical quality attributes and critical process parameters are exquisitely delicate features for mRNA-based therapeutics. So we've all learned a lot today about these features. Thank you. I'd like to remind our audience to submit questions. You can submit questions by typing them in the Q&A box, which can be found directly below the video player. Today, all questions received will be answered offline. So don't fear, we will be answering. There may be some small delays in certain cases. I want to thank the audience for attending and for participating in today's event. And I'd also like to very much thank our sponsor, Samsung Biologics, for making today's educational webcast possible. We would like to ask everyone in the audience to participate in a brief survey. This survey will populate automatically after today's presentation is ended. You'll receive an email alerting you when this webcast will be available for replay. We invite you to forward that announcement to your colleagues who may have missed today's live event. Thanks to everybody for joining today, and we'll see you next time. Goodbye.